Okay, I think we're good.
Great. It is my pleasure to introduce our speakers today, Dr. Chris Hurd and Dr. Tanya Bosak. They are the chairs of the Return Sample Science Working Group of Mars 2020. And so they will be telling us about sampling at Jezero Crater. So Chris, I'll turn it over to you first.
Great. Thanks so much for having me. It's a pleasure to be here and to give this overview of what we've been doing with the NASA Perseverance rover in Jezero Crater. And so I'll just launch right into it since I only have 20 minutes. The NASA Perseverance rover, Mars 2020 Perseverance rover, has four main goals. The first is understanding the geology of the region, the landing site. The second is astrobiology. The third is caching samples, and the fourth is preparing for human exploration. So we're going to talk really about those first three, and with an emphasis on the sample caching. The rover is very well equipped with instruments highlighted here. I won't go into all the details here, but suffice to say that we have a number of instruments with including spectrometry, both from the mast and on the end of the arm, that allow us to observe the surrounding, document the context for the samples that we take, provide geochemical information even at a distance, and then also up close when we're interrogating a rock and preparing to sample it. So Pixel and Sherlock, for example, and the Watson camera provide some of the detail that we need to be able to understand the rocks that we're up close. And this is a selfie taken by Watson after the first successful rock sampling here, that you can see the two boreholes down the lower left there, and our rover looking, I think, quite pleased with itself. Okay, we have landed in Jezero Crater, which is on the northwest corner of this impact basin, not far from the Nili Fosse region. This was chosen because, in particular, as observable from orbit, there is a delta deposit inside Jezero Crater, and this crater, which is about 45 kilometers across, was based on that observation, was filled with water, perhaps as much as 200 meters depth. At some point in its history, somewhere around 3.5 billion years ago, and the delta that was deposited there then provides a record of the sedimentology, the sedimentary processes that include transport of sediment down the inlet valley, which is known as Neretva Vallis, and the dumping of the sediment load from the river system into a standing body of water. And there's intriguing mineralogical sort of indications from orbit which have complement this observation, and you'll see some of that in a little bit later. Okay, we landed February 2021 at what is now known as the Octavia E. Butler landing site here on the crater floor. This is a traverse map showing you where we landed, where we continued. We drove south and dipped into this area known as Seta, and then backtracked and around this area, which was shown to be not traversable by the rover, along the crater floor, and then up to the delta front and across the top of the delta, and then into Neretva Vallis, which is where we are now. We've gone some over almost 28 kilometers total distance. And I won't talk about Ingenuity, but Ingenuity was a big success. It was the helicopter we delivered to the surface, and it flew ahead of us, and its final resting place is shown here in Neretva Vallis. The red dots here are where we have taken samples. Okay, and that's the main purpose here, is talk a bit about this. We have the capability of taking these samples, and the way in which we do this is we decide that we're going to interrogate a rock. In fact, we actually decide ahead of time that we're going to likely take a sample. And the coring mechanism, sampling mechanism, is in this cylinder on the end of the arm and in the belly of the rover. And in the bottom of the belly of the rover are a number of empty tubes where, and those are loaded into the sampling mechanism. But even before we do that, what we do is we put what's called an abrasion bit, and we abrade a patch that's 5 centimeters across. You can maybe just make it out in the shadow here of the arm on this particular rock. There's a 5 centimeter patch here, and that's in order to be able to clear a space on the rock beneath the outer rind, weathering rind, that to get at the fresh surface, and then interrogate that surface with pixel and Sherlock instruments on the end of the arm, as well as other instruments on the mast like SuperCam, in order to get at the mineralogy, petrology, geochemistry of the rock. And then, as you can see here, we swap out for the coring bit, which is in the center here of between these two stabilizers, which we place against the rock, and then we core the sample. And as we core the sample, it goes directly into an empty tube automatically. And then that tube gets processed and sealed in the belly of the rover. So the tubes look like this at the top here. This is one that shows with the sealed cap on the end here. They're made of titanium nitride, and they give us a core that's about 5.5. Usually it's about, actually, we run sort of between about 4.5 to 6, or even more than that, centimeters long and 1.3 centimeters across. And loaded on board is we have 38 identical sample tubes, and then 5 witness tubes. I'll come back to those. And we've used 27 tubes so far: 21 rock samples, 2 regolith samples, an atmospheric sample, and 3 witness tubes. So I want to talk about what we've done and where those are across the mission, and a bit about what we know about them in my talk. So we've completed 3 campaigns, and we're in the midst of a 4th. So the campaigns are set up in areas of interest, sort of subdividing the landing site on the basis of what is inferred or known from previous mapping. So this is a map showing effectively the same traverse that I showed you, although we've gone a little bit further since I put this diagram together, superimposed on a geologic map of Jezero Crater after Stack et al., so prior to landing. And so these different colors, of course, represent different geologic units as mapped from orbit. And so you can see the crater floor here has these 2 units, which we then renamed Maz and Seta. And the crater floor campaign is it constitutes this area here that includes the landing site and several other locations in and around Seta, the sort of thumb of Seta, as we nicknamed it. And then the delta front, the upper fan it's called, and then the margin campaign, which is where we are currently. And again, just like before, instead of the sort of red balloons, we have red crosses for where we have taken samples. Okay, just a bit about how we collect the samples. The sample priority is guided by community desires. And there's a report from 2018, extremely detailed report that looked at the potential science and engineering value of samples delivered to Earth by Mars Sample Return. And we use that as a guide. Many of those of us who are involved in the mission, in the capacity of return sample science, were co-authors of that report. So we kind of know what we're looking for. So those are the general Mars sample return desires. There's the Jezero Crater geology from the rover investigations. And then there's practical considerations that usually the engineers tell us whether we could actually sample something in a spot where we want to sample it or not. So all of these go into the samples that are acquired. Now, we don't just acquire them in a vacuum, of course. We acquire them with lots of essentially field notes. And what we have done is that we have set up a standard set of observations. And this stop list, standardized observation protocol list, is essentially helps to maximize efficiency and consistency. It includes things like the imaging of the workspace from the various cameras on board, remote science from, say, SuperCam, proximity science from the instruments on the end of the arm, et cetera. And then we record these in what's called a sample dossier, and that links to all the raw, basically the raw data observations. as well as things like where the rover was, the time history, et cetera. But the other thing that we do, and this is something that, again, those of us involved in the return sample science aspect of the mission are responsible for, is completing these initial reports. So for each and every sample, we have a templatized narrative description of the sample meant to be written within something like 3 weeks of collection, and it includes preliminary interpretation. It's not revised, it's preliminary interpretation. And so the first 21 samples have been fully documented in initial reports available on the NASA Planetary Data System. And so you can go, for example, to this link shown here and look those up and see what they look like. But essentially, it includes things like the workspace, sort of images like you see on the furnace piece here of the actual where we took the samples from, as well as the preliminary sort of raw data from the different instruments like Pixel and Sherlock, giving preliminary mineral identifications and texture. Okay, we also have these witness tubes. I'll just mention these. These are otherwise identical sample tube, but they're filled with different sort of getters, witness materials. And you can see an image from what's called cache cam. Cache cam is the camera that we use to look inside a tube before we seal it up. And we've done this several times now, but the idea here is that these different types of materials that are preloaded in this tube, they have a seal and then gets punctured and activated. And then in fact, it gets loaded into the end of into the sampling system. And we do everything except put a sample inside of it, including going right up against a rock. And the idea is that it captures some of the volatile material that might be coming off of the rover, because it is a machine, and it's, it does presumably has sort of off-gassed different compounds, including potentially organic compounds over time. And we want to have a record of that so that, when we bring samples back, we also have a witness tube that can tell us what might be coming from the rover itself, not from Mars. Okay, so the crater floor campaign I'll just touch on briefly. What we were able to do here is to map out a series of what turned out to be lava flows effectively in the Maz Formation, and sitting on top of, or stratigraphically above, the Seda Formation, which it consists of olivine cumulate rocks. So two different types of igneous rocks effectively. And I've shown here where we've taken samples. So the take-home message here is that we have this context of a number of lava flows interpreted from the rover instruments, and then within that we have a series of samples. Now the one in dashed line here was our first sampling attempt, which didn't actually get anything inside the tube for reasons that we now understand, but we actually count that as an atmospheric sample. So it's not actually a sample of the rock, but it does provide a Martian atmospheric sample. If we look at, for example, I'll just go back here. If we look briefly at this area at Brock, this is to give you an idea what it looks like. It turns out to have some layering in it, but when you look up close, it's filled with olivine, and the texture is consistent with a cumulate igneous texture. So this is quite intriguing that we would end up with sort of an igneous rock that has that sort of layering. But Pixel shows these really beautiful diagrams as shown in the lower right here, where the texture is very typical of this sort of igneous rock. So what we can say then from the crater floor is that we have a series in the Maz Formation, sort of on the outer part here, we have a series of lava flows of basaltic igneous rock. And these also have varying degrees of alteration by water that's percolated through them. So they're not completely dry, they have alteration as well. And then in Seta, we have an olivine-rich cumulate igneous rock. Itself also has evidence for alteration by water at some point. Again, we don't know exactly when this occurred, but there was water percolating through these rocks during their history. And just to give you a sense of what we're looking at here, for each of these spots where you can see where we have the sampling, if you look here in the top middle, alfalfa is the abrasion target. So this is the 5 centimeter patch that we took, that we analyzed to see what the rock was all about. And then Hahoni and Atza were the two samples that we took. These are the cache cam images. These are the actual cores that we took, looking down into the core tubes before we sealed them up. So in this case, we took 2 of every sample before proceeding along. Okay, then we moved on to the delta front, and Tanya will talk a bit more about this, rocks from this area, but just to give you an idea of what we collected here, for the first time we collected sedimentary rocks with the intent to bring them back to Earth. And so that's the most significant thing here, is a series of sedimentary rocks of different texture, but we also just mentioned this as well. We also, oh, sorry, this is the on the ground view of this area called Hogwellow Flats, and we took a sample up here at Skinner Ridge, a sort of more coarse sandstone and a finer grained siltstone down here, and mudstone down here at Wildcat Ridge, just to sort of give you a sense of what it looks like on the ground. And this, without going into detail, the main take home here is that because these are sedimentary rocks and because we have an idea of the stratigraphy, we have context. And that's really key for these samples, is we've collected samples within context, within their stratigraphic context. And that's very powerful. Okay, we also have the first regolith sample, and this is taken from this bed form that you see here in this location. And we use a special type of bit to collect this loose material, this regolith material. And so we took 2 samples from this spot as well. And instead of having an abrasion patch, we have a wheel scuff that we analyzed with the instruments to get an idea of the sort of original surface and sort of beneath the surface, to get an idea of the composition of grains, the distribution of grains, that sort of thing, prior to taking a sample nearby. So that's the first as well. Upper fan is a series of rocks that are, again, very intriguing sedimentary rocks, probably related more to the river system that was feeding the delta. There are rocks that look like this, as you can see in the upper right, these sort of curvilinear that we can see from orbit, stage of the fan development produced by the flowing river. And then this one here, Otis Peak, is a fluvial conglomerate. And this is really intriguing because it's a little difficult to see here, but there's a clast here that's centimeter scale that we can see in the abrasion. The abrasion behaved quite differently in this case because it's full of sort of centimeter, sub-centimeter clasts, or almost like rocklets. So you can think of, so within the scale of the sampling that we did, this is the cache chem image. This is 13 millimeters across. So you can see several millimeter sized in diameter grains that we see here, that once those are back on Earth, we could actually get a sense of the variety of rock types, igneous and otherwise, that were in the watershed for the fluvio-lacustrine system. So this provides a very powerful tool to essentially get small samples of a wide area that was eroded and carried into the river system some 3.5 billion years ago. Okay, so that's the upper fan. And then recently we've been in the margin campaign, and this was identified by the signature from orbit of carbonate, so iron magnesium carbonate within these rocks. And we've confirmed that. What we haven't confirmed is exactly what the heck this rock is. So there's still a lot of work that needs to be done to try to understand exactly how this was deposited. It still remains a bit of an enigma, and the team's working through that. So we collected 3 samples from within this margin campaign, and then we are currently right in this area here, which is known as Bright Angel. We just got some really intriguing data down from these rocks here, and that represents our likely I'll just say possible fourth sampling site from this margin campaign prior to moving on. So what do we have? We've collected 27 tubes, including 3 witness tubes. We have 16 tubes left, including 2 witness. If you notice, we were double sampling early on. We took 10 tubes, including 1 witness, so 9 sample tubes, and put them down in an area called Three Forks. We put the duplicates down essentially on the surface of Mars in an area, nice flat area in front of the delta, as insurance in case something happens to our rover later. So we have representative samples from the first part of the mission, essentially from the crater floor campaign and the delta front campaign we've set down. but we have duplicates of all of those samples that we've taken that remain on board, to which we've added singletons since then. So we have added, so you can see here, anything that doesn't have another name next to it that's on board Perseverance in the left-hand column here is a singleton. And so that includes everything on the fan top, and then these most recent three that are from the margin campaign. And so the idea, so to summarize, we have samples that are quite diverse. They include both sedimentary from lake, river, flood sort of environments, and igneous samples, multiple habitable or potentially habitable environments. We have samples both that include both inside Jezero and outside Jezero lithologies through what I mentioned about that. For example, the conglomerate. Different types of sedimentary rocks. We've sealed all the tubes nicely, and we are averaging something like 5.3 centimeters. And we're excited to continue sampling on the rim and beyond to increase the diversity of the samples. And the next thing what we're doing is what we've actually just finished the process of planning what's called the crater rim campaign, which is to drive up onto the crater rim and just beyond that. And from orbit, we see lots of variety in terms of the rocks that are potentially achievable from that location or from this area. And I would say the hardest thing that sits ahead of us is deciding which rocks that we are going to use to fill up the remaining essentially dozen sample tubes that we have, because there are way more lithologies, we think, than the sample tubes that we have left. So I will leave it there and turn it over to my colleague Tanya to tell us more about the sedimentary rocks that we've collected so far.
All right, so I'll try to share my screen. Here we go. Thanks, Chris. Chris gave a great introduction to how we are sampling, what we are doing with the rover, where the rover went. And I will tell you about, to at least people like me, the most exciting part of the cache that we acquired, which are these sedimentary rocks. The first sedimentary rocks that were deposited by water and altered by water that any rover has sampled, and the first rocks that have the potential to come to Earth from Mars. And so what can these rocks tell us about the origins of life and early life? Because this is really the reason why we are so curious about them and why we are going through all of this to sample them and bring them back. So why go to Jezero Crater? Chris showed you the fin and talked a little bit about the chemical composition and the minerals that are present. And so when we think about early life or prebiotic processes, when we think about whether there's ever been life anywhere else other than Earth, we try to think, really, we have to go far, far back in Earth's past. And so this is the timeline I think about when I think about time. I'm a geobiologist. I think about time in billions of years ago. And so this is the time scale in billions of years ago, time before present. And when we think about the first signals of life on Earth, even as early as 3.4, 3.5 billion years, for as long as there have been sedimentary rocks that we have preserved on Earth, we actually see signals of life. And this is what's indicated. And in terms of fossils, in terms of microbial mats, we actually see cellular life. So if you want to go even further back in time and ask how that life came to be, really, we don't have much material on Earth. The rocks get eroded, the rocks get subducted, and we really don't know what happened. And because Mars has these rocks that have been sitting on its dark and, well, really cold surface for billions of years, the hope is that maybe some of those rocks can take us back to this time that is here indicated by the dashed line and help us look for life elsewhere, or really tell us how early planets become habitable and maybe at least establish conditions that can harbor life. So this is what this landing site, the Jezero Crater, is supposed to cover. And this timeline here indicates really the span of ages. We, at this point, don't really know what those ages are, except that maybe the crater itself is about 3.8 billion years old. So the sediments are somewhat younger, the sedimentary rocks. But one of the major objectives for the rocks that we bring back is to figure out how old they are. So geochronology. And that helps us put everything else in the context of time. Okay, so talking about Jezero and talking about all of these rocks and minerals there, Chris already touched on the vein. That is a collection of sediments and sedimentary rocks that were delivered by either some gravity flows or water flows through this channel from the crater rim deep into the crater that at one point harbored a lake. So there's this inflow channel, there's also the outflow channel. And from the orbital measurements, all of this is done from the orbit. From the orbital measurements, not only did we see the fan, but also could detect some of the signals of minerals that made people really excited about the potential of these rocks to preserve some signals of maybe past life or at least organic compounds. And so Chris already touched on some of these. So there are some signals, so some of these purples will be signals of clay minerals, some bluish signals, but also what really got people excited and what makes Jezero quite a unique crater. There are other craters with fins, but this crater has this really intense blue and green band right here at the margin of where the lake would have been that is composed of carbonate. And from the orbit, when we looked at the signals, it seemed like these minerals here, carbonates that typically precipitate from watery solutions, these carbonates, unlike on Earth, where we have limestone, calcium carbonate, these on Mars looked like they would be magnesium carbonate and they would contain a lot of water. So we have carbonates, minerals that include carbon in them. So something that may have come from the atmosphere and the surface. There are also some patches really localized of silica. There are clay minerals, and all of this diversity excited people because this is exactly where we go on Earth when we look for early life. And so this band of carbonate that's really intense and hugging this area that would have been the lake's margin excited people because today there are modern day magnesium carbonate deposits. For example, there is this lake in Turkey, and this is what it looks like. So we really see a number of different sedimentary rock faces. So really these different types of rocks, including these microbialites, where actually most of this carbonate mineral is deposited by microbes. And then there are these terraces really close to the shoreline and the shoreline sands. And this is something that really was thought to be a good analog for what we may find in Jezero. This is why people said, yes, let's go there and let's sample this margin. We'll see. We'll mention what we actually ended up seeing. Of course, the idea here was also, and the great hope was maybe we can see something that is macroscopic and also bears signals of life, such as microbialites, because microbes can help precipitate these types of minerals and also make different shapes that you don't even have to have tiny microscopes or anything to sometimes even see and recognize a rock as something that was made by biology. And so in terms of microbialites, in terms of carbonate structures, we have a lot of such rocks from the early Earth, starting at about 3.5 billion years ago. And I'm just showing you this as an example. So these are some 3 billion year old microbialites, so laminated, actually stromatolites, laminated rocks. And just by looking, I won't go into any details, but just by looking at these rocks in the field or cutting them and looking at the thin sections, these layers upon layers, and looking at their shapes, you can tell that they were built by microbes. We can even start providing quantitative evidence for microbes self-organizing, organizing in the presence of light, looking at textures of these rocks. So there's a lot of hope for carbonates. Carbonates are great. There's also silica, which is really the dream if you are a fossil hunter in a microbial world. And what I'm showing you here is a 3.5, almost 3.5 billion year old microbial mat that was preserved in silica. So in fact, when people ask for the earliest signs of life that are uncontroversial on Earth, this is a picture that you may see. And what we have here is a thin section that was cut out of a banded shirt deposit of hydrated silica, actually mostly quartz nowadays. And when you cut it and put it under the microscope, what you see are these, so this is the scale bar, about 20 micrometers. We see a bunch of these filaments. So silica is really good because it's in preserving microbial shapes. So these are entire fossils of some ancient stringy filamentous microbes that are assembling and making, really intertwining to make layers of microbial mats. So for as long as we can go, we are fortunate to find silica and find microbial fossils. This is on Earth. So we were hoping to maybe encounter some of these patches of silica on Mars as well. And here are some of the features. And I'm showing these scales to give you a sense for what we could maybe expect to see. So if this is 20 microns, we certainly could see distributions of layers that are maybe slightly thicker using the rover instruments. Now, we also need to get lucky because most of the time when you look at charts, you don't find much even on Earth. And then the fan itself, with its abundant clay minerals and abundant evidence of the deposition by water, also gave us hope because of some discoveries by the Curiosity rover, our sister rover, in a different former lake on Mars, where here you see Curiosity exploring some mudstones, so rocks that were really once mud, because Curiosity in these rocks found some evidence, and some other rocks on Mars found some evidence for indigenous organic compounds on Mars. And so even if there's no life whatsoever, we still want to understand what the organic processes, was there any organic chemistry happening on planetary surfaces? Were meteorites maybe delivering organics? And to do that, once the samples come back to Earth, we first need to know whether the rocks contain organics in the first place and whether these organics come from Mars. And so this was another hope to find something that is fine-grained, that contains maybe some clay minerals, and has the potential of preserving organics, because the clay minerals and fine-grained rocks are what people on Earth, again, go to look for organics, actually to extract oil from, which is former life. All right, so all of these samples and Jezero as a potential site where all of these types of rocks may be found was of high interest to collect samples from, because if you're an astrobiologist and have all of these questions actually can be answered using these types of samples. So was there life on Mars? Then how long did the Jezero Lake persist? Basically what we are asking is for how long was there water in this system? And for how long was the system habitable? For how long could there have been some setup where life could have established itself? And to get at that, we also need to know what the atmosphere was doing. So by looking into all of these types of minerals, in particular minerals that contain structural water and carbonates, we can also get at what the atmosphere was like, because the atmosphere needed to be thicker to support water on the surface of Mars, unlike today, where there's basically no liquid water at its surface. And so if these are the questions, and I mentioned something about the ages of the rocks in Jezero crater, at least estimated, there is the potential to get at all of these questions by sampling Mars. And so here are some of the units. Chris mentioned all of these units in particular. So crater floor, the fan, the margin, and then the outside Jezero parts. So in terms of, again, look, if you look at these numbers, there are huge error bars, but this is exactly one of the reasons why we are going and want the samples back so we can date them. But if we go from Niliplanum, this is this outside Jezero part. So this is the higher elevated part in which crater was excavated. This is the oldest part. The margin itself could be as young as maybe the sediments, could be older. The fan itself probably younger than, actually we didn't know, than the crater floor, certainly younger than the outside of the Jezero. And the crater floor looked igneous to us. Chris now told you it is igneous. And we, again, don't have a really good estimate on its age, except that it's quite old, about from 2 billion to 3.5 billion years old. Okay, so here is our sampling path from this igneous crater floor over to the oldest sediments that were deposited by water at the front of this fan. And this is really what I'll be talking about. I'll highlight the specific sample from there. And so if you're looking for biosignatures, or if you're looking to understand habitability, it is really this region on the fan, but in particular this region that is the oldest part of the fan that is of particular interest, because this is really getting at the earliest conditions of habitability and also potential for life. And then we continued on to the margin, this carbonate-rich area, and now we are almost on our way out of Jezero. And so here, this part is igneous. I won't talk about that. Chris showed you some samples, and I'll focus really on the samples here from this part that is at the front. And in particular, one of the samples that is coming really from here, the finest grained of sediments. And again, just based on the Earth analogs, I mentioned those, and also Martian analogs, I mentioned those mudstones. We need something that is quite fine grained, not porous, to preserve organics and to preserve biosignatures as well. You need a lot of tiny silica granules if you are to preserve a microbial body fossil. And these granules have to be nanoscopic. Therefore, we are looking for something that is as fine grained as possible. And so what we found, in fact, on the fan itself was not that. We did not find mudstones. We mostly found sandstones. We were really pleased to see that a lot of them contained carbonate. We also found silica in some of them, but we did not find, as we were hoping, something that would be very similar to what was observed in Gale craters, such as these really fine-grained mudstones. But what we did find is one mudstone that was present. That was that bright area here. Sorry, here, this point here, really at the very front of this fan. And this is what it looked like. So the rocks themselves were quite bright, and they looked quite flat, somewhat fractured. And so when we looked at them from above, here's the abraded patch. Here's also, this was taken after we already cored the sample. What we found out by looking at the abraded patch, here it is you actually see that the rock is quite fine-grained. So this is the scale of 1 centimeter. You see some grayish areas, you see some reddish areas. It turns out when we brought our instruments close to this abraded patch, and these instruments are Pixel, which does X-ray fluorescence spectroscopy, so it tells you what elements are present, and also Sherlock, Raman, and also UV fluorescence, which tells you, so the Raman can tell you about the presence of specific minerals and water. What we found was this rock is actually made of iron magnesium clays. So yay, clay minerals, good potential to preserve organics. And also we found that the rock was cemented by these iron magnesium sulfates. All iron is iron 2, and also fine grained, which was somewhat unexpected, indicates conditions that contained water once. Probably very salty conditions, because iron magnesium sulfates really need a lot of water to go away before they precipitate. But all in all, this is really the sample. We're excited because this should be the sample with the highest astrobiological potential, with the most potential to preserve organic materials and any potential biosignatures, such as maybe microbial fossils or even signals of microbial mats, if anything was present. Now, we did not find mats here. We did not find anything. Our instruments are really not good enough to go to the level of individual or even aggregates of microbial fossils. But when we started looking at the data from there, we got very excited because we saw a lot of fluorescence in these. So these rocks are great for biosignatures. There's also water present, so we can one day study this water to understand how the atmosphere was lost from Mars. But we also initially got excited thinking there may be some organic materials present in these rocks. And this is why. This is really just learning how one uses the instruments and how one analyzes rocks in real time on Mars. So the instrument Sherlock measures ultraviolet fluorescence, and this is our chief way of detecting organics on Mars. It does so in spots that are maybe 120, 150 microns in diameter. So again, much bigger than individual microbial cells. And the idea is the following. So if this instrument shoots a laser beam, UV laser, at the rock, at this abraded surface, in a raster, over a patch, and detects the fluorescence that comes back from the rock. And if this fluorescence is detected in particular windows, such as here, that may be matched to some organics. For example, here are a bunch of, so these were analyzed in the lab, bunch of aromatic organic compounds that seem to emit in this window from about 305 to 350. This is what their fluorescence looks like once you shoot laser beams at them. And sure enough, so this is the Earth, and then Sherlock came to these rocks, these really particularly fine-grained rocks that are sulfate-rich, made the measurements, and look at that. It's really, we are detecting fluorescence exactly in the same part of the spectrum. However, Eva Scheller, who is a postdoc actually working currently at MIT, and was one of the earliest Sherlock people even developing standards and measurements to understand the detections of organics and minerals, started worrying about any potential inorganic signals, something that we have to worry about as astrobiologists. What are any possible abiotic signals that could produce the same types of measurements as organics would? And so she started worrying about trace inclusions of elements we can't really detect with any other instruments. For example, cerium, that is a really common inclusion, common trace element in phosphate minerals that really has fluorescence. So here is a bunch of curves of cerium bearing phosphates, just slightly different phosphate mineral compositions. And you can see some of them match this, this fluorescence really well that we observed. Really depends on the kind of mineral specifically that you measure. And while we can detect cerium, it occurs at parts per million in rocks. There are no other instruments on the Perseverance we can look at cerium with. It may be that cerium and phosphate or sulfate really start producing very similar fluorescence. And so the question to ask was, because we are looking here, astrobiologists, we really do want to know whether there's organic matter in the samples. Our Raman signals really don't show much. And so Eva decided we can't really confirm it's cerium, but we may see that if really this fluorescence has a distribution, that the distribution may match the distribution of phosphorus. And so here is a surface of a rock that we abraded, shown in visible light, and then Sherlock comes and makes a map, measures luminescence in a number of pixels over that same area, and here are the luminescent spots that were initially interpreted as organic. And then Eva correlated that map with the map produced by another instrument that just measures phosphate. And look at that beautiful agreement. So to the first order, and again, this is something that every astrobiologist really needs to worry about. We need to worry about abiotic sources of our signals. And to the first order, as a null hypothesis, phosphate and cerium in phosphate seem to be the most likely explanation for what you're seeing. And so the good news is now we, I think, really have a more stringent set of criteria by which to recognize real organics if we were to one day see them. And so these rocks have to date the highest astrobiology potential, organics or not. We still, so not all of this, what I need to mention here, while I'm saying here we are not detecting them, our rover instruments are highly insensitive to organics. This is also in thinking about all of this, what we realized is you would really need a lot of organics, something that matches the abundances in really some quite good terrestrial rocks to see anything. And any lower abundances, while we can detect anything, if organics are present, they would be no problem to detect with the Earth instrument. So if you extract the rocks and you try to look for organics by mass spectrometers on Earth, that should be no problem for detection. People are detecting much lower quantities. It is just that we can't pack these mass spectrometers on the rover. And again, this is why we need to bring these rocks back and really look for organics. All right, and so another point, I was talking a lot about carbonate, and so while Chris talked a little bit about the margin, so here are some rocks from the margin. I said there are carbonates in all of these rocks on the fan. These are sandstones. It's really, if you're looking for fossils, sandstones are not what you will probably sample, because it's just really the energy required to deposit sand grains is not really conducive to microbial colonization at most times. But here we did see in the margin some rocks that contained pure carbonate grains that were cemented by silica. So while we don't know how these rocks really formed, and we don't quite understand the depositional environment, it may have been maybe a lake margin, but maybe it was something completely different. These rocks importantly contain two materials that we were hoping for initially. So both carbonate, that can tell us quite a bit about the atmosphere, can tell us quite a bit about the solution chemistry in which carbonate precipitates. And also we see silica cement that actually encases all of these carbonate grains. And remember, silica, I said that's the dream for any fossil hunter. So one day, if we get to analyze these rocks, I am sure we will be surprised by what we find in there. And so this is really another sample that I think is incredible and could preserve biosignatures, certainly will tell us a lot about the fluvial activity, the water activity in Jezero crater. And with all these hydrated materials, there's quite a bit of water in the silica. We could get at how the atmosphere was lost and for how long the water was there in Jezero. So I described just two samples that we collected, and I hope you realize how they can be used to start getting it for the first time, really. We have the potential to start understanding the answers or really asking these questions using samples from Mars and using samples that are older than anything that is present on Earth. And again, so here, if these are the samples, so here are the samples from the very fan front, these are the samples from the fan top, and maybe from the margin, maybe some of the margin ones are actually older. We have really the potential to span all of these times in the deposition of the fan and the times that to capture the entire interval, while water was present on the surface of Mars during the deposition of the fan. And so just to summarize, the rover has acquired 20 oriented cores of bedrock. So there are certainly regolith samples that you can ask Libby about, and the atmospheric samples. These are the first samples of extraterrestrial sedimentary rocks from any planetary body that were deposited by water. So something to really appreciate as a time, as samples from a time when Mars looked very, very different than it does today. There's no clear evidence yet for organic matter of fossils, a lot of sandstones, and most contain carbonate. This mineral that does contain carbon, potentially signals of the old atmosphere. And after a return to Earth, these samples will revolutionize our understanding of Mars and its early habitability and potentially even the evolution of life. So I will stop here and then